Today we will be discussing the regulation of phosphofructokinase, which is a very important enzyme in the third step of glycolysis. It turns fructose 6-phosphate into fructose 1,6-bisphosphate. Now, phosphofructokinase is a very important enzyme that requires regulation because this step is a committed step. Once you have made fructose 1,6-bisphosphate, you must proceed through with glycolysis. Like this molecule can't do anything else. So, and this is also a very exergonic step, and so it has to be regulated. So the regulation starts with hormones, glucagon and insulin. We'll talk about glucagon first. So glucagon is released by the pancreas when there is low blood glucose levels. It ultimately results in CAMP production, which phosphorylates a multifunctional enzyme. This thing right here represents the multifunctional enzyme. A multifunctional enzyme is basically an enzyme with two domains, and depending on whether it's phosphorylated or not, determines which domain of this protein will be active. So when it's not phosphorylated, you can see that the PFK2 domain is active. And when it is phosphorylated, the FBPase2 domain is active. So the way this works is when there's a lot of CAMP, it is going to phosphorylate this multifunctional enzyme. When it's phosphorylated, we can see that the FBPase2 domain is active. And FBPase2, right here, you can see it it removes a phosphate group from fructose 2,6-bisphosphate. That's what phosphatases do. They remove phosphate groups. So when it removes a phosphate group from fructose 2,6-bisphosphate, it essentially stops glycolysis from happening. Why? Because fructose 2,6-bisphosphate actually activates phosphofructokinase, and it activates phosphofructokinase, which means it stimulates glycolysis. Now, if the concentration of this is decreasing because FBPase2 is converting it into fructose 6-phosphate, then the glycolysis will slow down. Now, the opposite is true for insulin. Insulin is the hormone that is present when there's a lot of glucose in the blood. And what it does is it stimulates glycolysis by increasing the concentration of 2,6-bisphosphate. How does it do this? Well, insulin is going to activate the phosphoprotein phosphatase. Phosphoprotein phosphatase. What it does is it removes the phosphate group from this multifunctional enzyme. When it removes the phosphate group from the multifunctional enzyme, the PFK2 domain becomes active. When the PFK2 domain is active, it adds a phosphate group to the fructose 6-phosphate, which results in higher concentrations of fructose 2,6-bisphosphate, which can go ahead and activate our phosphofructokinase and allow glycolysis to resume. Or in other words, it stimulates glycolysis. So, summary, glucagon will, the presence of glucagon will inhibit glycolysis while insulin stimulates glycolysis. Glucagon inhibits glycolysis by increasing the CAMP concentration, and when you increase the CAMP concentration, what you do is you activate the protein kinase. And protein kinase loves to phosphorylate stuff, so it's going to phosphorylate the multifunctional enzyme, which will mean that the FBPase2 domain of the enzyme will be active, and the FBPase2, whenever that domain is active, what it does is it removes the phosphate group from this fructose 2,6 bisphosphate, and the decrease in the concentration of fructose 2,6 bisphosphate ultimately results in not enough activation of phosphofructokinase needed to continue with glycolysis. So this is how this very important enzyme, phosphofructokinase, in the third step of glycolysis is regulated. Thanks for watching.